Okay, good morning, everybody. Every class was stopped in a slight different part yesterday with the whole projector thing that went on the other day. The afternoon classes are a little bit behind, so I'm just going to start here with just the very tail end of Chapter 12. So if you're in a morning class, first or third hour, you're past this, so you can maybe fast forward this a little bit. So for everybody, if you remember, we were talking about cancer and how it relates to mitosis, and we talked about what types of things can mutate or change the DNA and therefore cause the cell to become cancerous, and that's called transformation. So I think all classes got to that. And then what genes are mutated to cause cancer, and I think all classes we talked about these set of genes called proto-oncogenes. So you can pause this and write this down, and I'm going to continue to talk about it. So these are normal genes that stimulate normal cell growth and division. So this is in everybody, and it helps for your cells to divide when they need to divide. But what happens is if one of those carcinogens or radiation mutates your DNA, and the one cell in the DNA that's mutated happens to be a proto-oncogene, that mutation can kind of increase the product of the proto-oncogene or increase the proteins that stimulate cell division. So you get even more cell division, which is what cancer is. So mutation to one of those genes, notice that there's an S on there, so there's multiple genes like that, and that can lead to cancer. The second type of gene, if it's mutated, that can lead to cancer is called the tumor suppressor gene. These are also normal genes in your body, and their job is to inhibit or stop cell division when it's not supposed to go on. So that's the normal job for it. So you can imagine if these are mutated, then the cell division isn't going to stop when it's supposed to, which also leads to uncontrolled cell division. So another mutation to one of these genes can ultimately lead to cancer. And so that's your last class there. And I just want to talk a minute about how cancer cells differ than other cells, and that they divide, kind of the loss of the cell cycle, and kind of divide uncontrollably. So normal cells divide between 20 and 50 times in the process of aging. I'm going to put here in the word some in front of here. Some cancer cells are seemingly immortal because they continue to divide forever if they're given a continuous supply of nutrients. Not all cancer cells will divide forever, but they do divide way more than the normal amount, and therefore you get an excessive amount of them. So they don't behave normally due to this loss of cell cycle control. But there have been found cancer cells that have been seemingly immortal, and an example is the HeLa cells. So Henrietta Lacks, that's the name where HeLa comes from, He from Henrietta, La from Lacks, had cervical cancer, and cells were taken from her when she went to get treatment. They tried to save her. She did die, and the cells that they took out of her and were cultured in the lab, so they're growing in a petri dish in the lab, and this was in 1951, and still today there are many, many cells from her culture, and they've been growing, and they've since mutated, and there's different strains of them, and there's been lots of research done on them to help with the fight against cancer and so on. And the thing is that she didn't know and hadn't given permission to do research on the cells, so that was all done without her knowing it. But they're a little unique in that they have been dividing since 1951 and have been alive and so on. So obviously they have exceeded the normal 20 to 50 cell cycles. So that's just a little thing about Henrietta Lacks, and lots of research has been done on her cells. So for, let's see, fourth and sixth hour, I don't think we got to this, and I have just a small little video clip that I'll show you on Monday that is about the HeLa cells. All right, and so that is chapter 12, which was all about mitosis. And so I'm going to do a little thing on this, just a blank sheet of paper, linking mitosis and into meiosis. If you look at your chapter 13 packet, notice the beginning of it. I start with an intro to genetics, 14.1, and that's because meiosis is the making of egg and sperm cells, and that has to do with genetics, because that's how you give your traits and your genes to the next generation, is via the egg for the mother and the sperm for the father. And so meiosis kind of marries and goes right with genetics. And so I'm going to do a little intro to genetics and also relate it to mitosis here. All right, so we've talked about how draw here. You might want to get yourself a little piece of paper as well if you want to, or there's a back side to the chapter 13 notes. And so remember DNA is wrapped around proteins. And so here these histone proteins, here are the histone proteins, and DNA is wrapped around these histone proteins. And that DNA, remember, DNA is, if we just look at the DNA, has genes on it. So genes are a section of DNA, and gene A codes for making protein A, which will give you trait A, whatever that is. It could be a protein that helps you to digest protein in your stomach or something like that, so trait A. And then later on you have another gene, we'll call it gene B, contains the instructions to make protein B, which will give you another trait, trait B. All right, and this might be a protein that's a pigment that gets deposited in your skin to give you skin color. All right, and then, so a one piece of DNA can have many, many genes. It can have hundreds of genes on one piece of DNA, and we have 46 pieces of DNA to make up our 46 chromosomes. So we just finished up with mitosis, and so let's just do a quick little thing about mitosis. I'm just going to do an overview and relate it to this gene stuff. So mitosis, I got a couple colors here. We started out with a cell, and in the nucleus of that cell, you had chromatin, and you have two of every kind of chromosome. Remember when we did the karyotype? Two of every kind of chromosome, one that you get from mom, one that you got from dad. And so before cell division, the DNA, remember, is loosely packed in that chromatin form. So I'm going to draw here two pieces of DNA. Let's say this is chromosome pair number 1. All right, this is DNA, this is pair number 1. We would have 23 pairs. I'm only going to draw one pair because it's easier to do that, but you have to imagine that we could draw 23 pairs to give us a total of 46. So it's very loosely packed. And so what happens is, and I'm not going to draw every phase here in mitosis, but remember in the S phase of interphase, we still have a nucleus. During the S phase of interphase, what happens is the DNA is replicated. So what happens is it's still in the chromatin form, so this is the original piece of DNA right here, but then we make an exact copy of it right here. And so therefore, and it's attached at the centromere. So you have an exact copy of that DNA, and then the second strand, we make an exact copy of it like this, connected at the centromere. It's in the chromatin form. So this is two kind of pieces of chromatin. I actually could say replicated. And this is two just chromatin. It's loosely packed. Okay, so then let's fast forward a little bit here. And so as we go, this is interphase, and this is the S phase of interphase. Then remember the nuclear envelope breaks down, the chromatin starts to condense, the spindle forms, and eventually these condensed chromosomes, which are not called chromatin, they're called chromosomes, line up down the center of the cell. So then what we get from here is, here's your cell, this DNA is going to condense. So we have, I'm just going to draw squiggles here, and shorter and stockier chromosomes. So this part right here is this part right here, and then this is the exact copy of the DNA. And then over here, this one does the same thing. And I'm showing them lining up down the center of the cell. And remember the spindle attaches, all right, to each side. And then what is the spindle going to do? It is going to pull the chromatids. So what we have here, I should write here, what we have here are two replicated chromosomes. Replicated or duplicated. I'm just going to abbreviate C-zone for chromosomes. All right, so we have 2 replicated chromosomes, and so they are going, the chromatids, each chromosome is made out of 2 sister chromatids, the exact copy of the DNA, are going to separate to opposite sides of the cell. And so if we draw the next cell, this would be anaphase. They look like this. They're being pulled to the opposite sides of the cell. So 2 pieces of DNA. Here's your spindle fiber. And then remember that the cell is going to start to indent here and indent here. If it's an animal cell, that's the cleavage furrow. If it's a plant cell, it's a cell plate, and divide into 2. And then when it divides into 2, the nuclear envelope is going to reform, and the DNA is going to unravel and become loosely packed again and become chromatin. So this here is unravel. This one right here, unravel, and now we have back to 2 chromatin and 2 chromatin. And so notice here, we have exactly the same as what we started out with. We have 2 of each type. So the key is to replicate and have them line up single-filely, so you have 1 of each type at the end, 1 of each, and you have exactly what you started out with, 1 of each chromosome. And so you have a total of 40. If it was humans, we'd have a total of 23 pairs or 46 chromosomes. So we have pair number 1 here, and then we'd have pair number 2, 3, 4, all the way to 23. And so so this is real important because 46 chromosomes is the magic number for humans, and that's because that's what gives us the appropriate DNA. And why that gives us the appropriate DNA is because it has the appropriate genes on it. So how do these pairs relate? So when we say chromosome pair number 1, these guys are not identical, but they have the same trait on them. What do I mean by that? So you have 2 chromosome number 1. So remember, a piece of DNA has genes, can have up to 100 genes. And so when we say like right here and right here, this piece of DNA at that particular point, let's say you have a gene to determine if you're going to have freckles or not. And then right here and here, you have a gene that will determine what skin color you have, and so on and so forth. And so on each chromosome pair that you have, while these the genes are not identical, what trait that they code for are. So you have a skin color gene and a hair color gene and a freckle gene and whatever it may be in the same spot on these 2 pieces of DNA, and that's the case for all of the chromosomes. And so all chromosomes have different pieces of DNA. So when we go back to look at a karyotype, this is an actual real picture of a karyotype. Remember, you have 2 of every kind of chromosome. So the DNA that makes up the 2 strands of DNA for pair number 1 is different than pair number 2, and so on. But your 2 chromosome number 1s have the same traits on them, and they code for the same traits, but they don't have to have the same gene. So just because you have, let's go over here, a hair color gene here and a hair color gene here, it doesn't have to be code for the same hair color. It's just a hair color gene. And we call these chromosomes homologous. So these 2 are homologous because they have the same traits on them in the same order in the same spot, these 2 chromosomes. And then chromosome pair number 2 are made out of different pieces of DNA which have different traits on them than chromosome number 1, but they are homologous to each other in that there's the same types of traits on each one of them. All right, so we have homologous pairs of chromosomes, and that's how they relate to that. And so at the end, when you get the end of mitosis, you have 2 of every kind of chromosome still, 2 of every gene for every trait that you have. And that's what gives you the appropriate amount of DNA. So too much or too little creates issues. So leading into meiosis, meiosis is making egg and sperm cells. We cannot make egg and sperm cells by mitosis because then we'd have a baby that has 92 chromosomes because the mom would give them 46 chromosomes. Then the dad would give 46 chromosomes in his sperm cell, and the baby would have 92, and the baby would not survive. So at the end, to make egg and sperm cells, we need the cells at the end to have half the number of chromosomes, and not just any half of the number of chromosomes, but one of every kind. So one chromosome number 1, one chromosome number 2, 3, 4, 5, and so on and so forth. So that's what we're going to look at here over here. So let's look. So I did meiosis or mitosis. Now we're going to look at meiosis. So I'm going to draw a little line here. I'm just going to write here meiosis. Okay, so meiosis, we start out, the cell looks the same as over here in mitosis. So we start out with a nucleus, and you have, in the ovaries of females or in the testes of males, you have your chromatin, and here's chromosome number 1, and again, we'll just go through with one chromosome pair, but there's 23 pairs. And so the beginning starts out the same. So what's going to happen is we're going to replicate our DNA. So we have the replication of the DNA, still in the chromatin, which, by the way, let me go back over here, as I just realized I didn't write how many chromosomes we have here. So we said that this has 2 replicated chromosomes. We would say that these are 4 single chromosomes. And then here, once the cell divides and they unravel, it goes back to chromatin. When it's still condensed, because these are still condensed, they're considered chromosomes. All right, so I just realized I forgot to do that. So here we're starting out the same, 2 chromatin, 2 replicated chromatin. And then the same kind of thing is going to happen as in mitosis, is that the nuclear envelope is going to divide, you're going to get the spindle forming, and the chromosomes are going to condense. What differs here is how they line up. And let's think about what the end product is. These guys lined up single-filely so that each cell at the end got every kind of chromosome. We don't, in 2 of every kind of chromosome, so I got 2 chromosome number 1s. In meiosis, we don't want that. We don't want to have pairs of chromosomes. We want 1 chromosome number 1, 1 chromosome number 2, and so on. And so they can't line up single-filely, because if they did line up single-filely and did what mitosis did, you'd end up with cells with 46 chromosomes, and that would be no good. So what we need to do is have the chromosomes line up differently. And so, if I just fast forward a little bit, I'm not going to put all the phases in there, but so the nuclear envelope disintegrates, spindle starts forming, and they start to line up. Before they do that, though, they do something a little different called crossing over. And so this is where the chromosomes overlap. And so if we look at this here, I'm going to go back to here and have these guys overlap. So they have the little parts of the DNA overlapping there. So then what happens is they condense and line up. And so instead of lining up single-filely, they line up side by side with their homologous partner. So here's the condensing here, and here's this. But you see how they overlap here? They're still overlapped. They go like this. I know I'm making it a little longer there just to show the overlapping there. So they condense, and so they're lining up next to one another instead of single-filely. And so what happens is then when the spindle forms, it attaches only to the left spindle attaches only to one of chromosomes. So this is pair number 1, one of chromosome number 1, and then on the right side it attaches only to the second chromosome number 1, and so they're still crossed over here. And so, so what that's going to do is when anaphase happens, and the spindles pull the chromosomes to opposite sides of the cell, instead of separating chromatids like they did over here, this chromosome is going to go to this side of the cell, this chromosome is going to go to this side of the cell, and what happens is right here is where they crossed over, they swap DNA. And so I'm going to do 2 different colors here. So this part here, let's say this part was on this chromosome. I don't know if you can see that. And this part is on this chromosome. And so then they swap pieces of DNA. And we'll see later that that helps to increase genetic variation. So that during this crossing over process. And so what happens is, as we look over here, is that when the cell divides, this is going to go to the left and this is going to go to the right. And so it's going to divide into two cells. The cell on the left is going to have this chromosome with the new piece of DNA that was on the other chromosome. So there's a new piece of DNA there attached to it. And we'll go over that more in detail later. And this cell is going to have this chromosome here with a new piece of DNA attached to it. And so it's not identical these anymore because the sister chromatids. And so now let's look at what we just did. So we just went from our original cell had 2 pieces of chromatin. So if this is a human cell, it would have 46 pieces of chromatin. Replicates it. Now we have 2 replicated chromosomes in these cells, in this cell. And then when this divides in half, and you get 2 cells here, this has 1 replicated chromosome. And this one has 1 replicated chromosome. And it's chromosome because it's still condensed. So it's a replicated because it has made out of 2 pieces of DNA that are identical. So it's made out of 2 sister chromatids. And so then what happens is these cells can't be your egg and sperm cells because they don't have exactly half the amount of DNA that we had to begin with. So what these cells do have are one of these guys here. But they have 2 replicated pieces of DNA attached. So what we have to do is separate these guys. And so that's what the second part of meiosis. So this is called meiosis 1, where what happens is the homologous chromosomes separate into different cells. And what happens in meiosis 2 is that, if we go to meiosis 2, these chromosomes line up single-filely. This is very similar to mitosis. They line up single-filely down the center of the cell. And this time the spindle attaches one from each side attaches to the chromosome, because what's going to happen is your chromatids, each half is going to separate into opposite sides of the cell. And so what's going to happen is that in anaphase, you just draw that here. In anaphase, you have, so these guys are being separated. The side's going to this side, and then same thing's going on over here. The chromatids are separating. And the cell is going to eventually divide in half. It's going to divide in half, and the chromosomes are going to unravel. So we get 2 cells here, 2 cells. I'm running out of room here. 2 cells here, and this unravels and becomes chromatin again, chromatin, 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 and nucleus reforms around that. And so if you look here, it has 1 piece of DNA from chromosome number 1, exactly half of what we started out with here. And so so this is, we'd have, if we write here, the cell here, once they start to separate, this has 2 single chromosomes. And then this has 1 chromatin, all of these. This has 1 chromatin, each one of these. And these are your egg or sperm. So in humans, they'd have one chromosome number 1, one chromosome number 2, 3, 4, 5, and so on, so that when the egg is fertilized by the sperm, the baby will have 2 chromosome number 2s, 2 chromosome number 3s, and so on and so forth from each parent. So meiosis first separate so you don't have pairs, because you don't want to have pairs in your egg and sperm cells. And then the second thing, so that's meiosis 1, is separating the pairs so you don't have pairs. And then meiosis 2, so this meiosis 2, the second division of meiosis, reduces and separates the sister chromatids. So this separates the homologous pairs. This separates the sister chromatids, and we end up with exactly half the DNA as you did in your original cell. And so that's the special division for meiosis. So relating this to genetics, the whole purpose of making egg and sperm cells is to make to pass on these chromosomes to your kids, so that they not only have the right number of chromosomes, but they also have the genes that are in the DNA of those chromosomes, so that your kids have a combination of genes from both parents. And so I want to spend a minute and just review with you. I'm sure you remember a little bit about genetics, but review with you a little bit about how genetics and genes work, and then we're going to come back and relate it to mitosis and meiosis. So with genetics, you don't have this picture. I'm just going to go over a little review with you. I'm sure you remember, or it rings a bell, Gregor Mendel, considered the father of genetics, and he did a lot of experiments to figure out how traits are passed from parent to offspring and how that exactly works. And so he did a lot of work with plants. He was in charge of the gardens in a monastery. He was a monk, and he crossed and quote-unquote mated flowers to see what kinds of traits would show up in the offspring, and he did a lot of good work. So how he crossed the plants. So here he is crossing. Starting out, he called the first generation the parental generation. He crossed a purple plant with a white plant, meaning the flower color. And so how he did that, I've talked a little bit about this before. You have the stamen, which has the pollen, that's the male part of the plant, and you have the carpel, which is the female part that has the eggs. And so, so what he did to kind of make sure that he could cross the plants that he wanted to. He took the purple flower and he removed the stamens. That's the male part. They have the pollen that has the sperm. He did that so that they can't self-pollinate, because flowers, the pollen from the same flower can fertilize the eggs of the same flower. So flowering plants can self-fertilize, or they can cross-pollinate, where you have the pollen from a different plant can fertilize the eggs. And so that's what he wanted to do, but yet he wanted to pick which plant the pollen came from. So he transferred the pollen, which contains the sperm, from the stamen of the white plant and put it on the top of the carpel, which the sperm goes down the pollen goes down a pollen tube here, where the sperm goes down and fertilizes the egg. And there are multiple eggs down here in the carpel. And so different sperm can fertilize different eggs, and then you get zygotes, and then those zygotes are wrapped around in a seed. So what this ovary at the base of the flower develops into the pea pod, and each pea is a baby plant. So each pea had an egg, represented an egg that was fertilized by a different sperm. So therefore, these peas are kind of like brothers and sisters of each other. They're siblings. They have the same parents here, but were fertilized by different sperm. And so then you know the parents, that's the pea generation. And so then to see what traits, you wanted to see, okay, if I crossed a white and a purple flower, what kind of traits am I going to get in the offspring? So how to tell what kinds of flowers they're going to have is he had to plant these guys. So he planted these seeds, And he called these the F1 generation, the first filial generation. And he planted them and let them grow, and they were all purple. So I wondered, okay, so I have a purple parent and a white parent, and now I have all purple in the offspring. What happened to the white? So what he did is he took two of these flowers, all right, so took, kind of the siblings here, and crossed them. So he did a purple with a purple. And what he got, let me show you another picture. What he got is that when you cross so this is the P generation, purple crossed with white gives you all purple. And then he crossed purple crossed with purple, so crossed with another purple. Crossed two of the F1 generations. Their kids, he crossed them, planted them, let them grow, and he found that he got some purple and some white. In fact, he got a 3-to-1 ratio every time, of 3 times as many purple plants as white plants. But he began to wonder, why is that? Why is it that I had a white plant here and crossed with a purple, got all purple, so the white seems to disappear, but when I cross two of these, the white seems to reappear. So where did that white go in that middle generation? And as you probably may have guessed, if you remember from genetics and biology, the white didn't really disappear. It's just that the white is a recessive gene and the purple is a dominant gene. So what happens is remember, I said that when you have homologous chromosome pairs, you have similar traits, so the same trait, but different versions of the gene for that trait. And so that's exactly what happened. So what he found is that on the pieces of DNA for the purple flower, so you can kind of maybe draw this out, purple cross with white, is that on their original piece of DNA, let's say the purple trait flower is on chromosome pair number 1. And so remember that there are genes in various spots. So let's say the gene is right here. And so what he found is that these guys had 2 purple genes. Purple gene and a purple gene. And then here, the white flower on their chromosome number 1, in that same spot, there's a flower color gene. But in the white flower, instead of the gene being the purple gene, it was 2 white genes. So then when they cross these, remember when the purple flower and the white flower make their egg or sperm cells, they reduce the chromosome number. So going back to this picture here, if you had 2 chromosome number 1s, when you go through meiosis, you're only going to pass on 1 chromosome number 1 to the kids. And so it's a 50-50 shot whether or not it's going to be this chromosome number 1 or this chromosome number 1. So what happens here, though, is for these two, for the purple plant, all right, so if this is in their cell, when they go to make, let's say it's the purple flower, I don't remember which parent it is, but when it goes to make its gametes, it's going to give this chromosome number 1 or this one. It's only going to give one of them at the end. But in this case, for the purple trait, it doesn't matter because both of these genes are a purple gene. So this parent is going to give to every kid it has 1 chromosome number 1, and that 1 chromosome number 1 that it's going to give is going to have a purple gene on it. And then this parent here, when it goes, so here's the cell, it's going to go make its gametes. It's not going to give both chromosome number 1s. It's going to give just 1. And so this one, it doesn't matter which one it gives. No matter which one it gives, it's going to give also a flower color trait, but it's going to give the white flower color trait. So now this purple plant has a purple and a white gene, but guess what color it becomes? It only shows up. It only shows up as purple. And so that's what happened. And so the white doesn't show up. And so that's where he coined the term dominant and recessive. Which one would be dominant? Think in your head. It's the purple that's dominant. The white would be recessive. what the definition of dominant is when you have two different versions of a trait in one individual. So that's what we have, a purple and a white. The one that shows up is the dominant one. All right, and so the white one is recessive. It's hidden when it has a dominant trait. And so therefore now we can go to our fill in the blank notes and start to fill those in with a, so that's the first two vocabulary terms. So it says blank is the gene that shows up when two opposite versions of the gene are present. And then the second one says it's a gene that doesn't show up when two opposite versions of the gene is present. So the first one should be which one? Dominant or recessive? It should be dominant. And this is recessive. Now, when I was writing on here, about the genes, and I wrote purple and white. Instead of writing those words out, we use shorthand, and you probably remember capital versus lowercase letters. And so whenever you're looking at a trait, you can, instead of writing out the actual trait, you can pick a letter for that particular trait. So let's go over here. So let's say the trait in this case is flower color that we're looking at. And then our two different versions of the trait are purple and white. And so purple is dominant. And white was recessive, right? And so we can pick a letter to shorthand it. So in the dominant trait is represented by an uppercase letter, and the recessive is represented by a lowercase letter. So I'm going to use the letter R. So big R in this case would be purple flowers because it's dominant, and a little r would be white flowers because it's recessive. And so but the thing is that when you're, for one trait, your two letters have to be the same. So it has to be an R for the dominant and recessive. You can't have a big R here and a little T and have it be for the same trait. So then another, if you did plant height, you could pick another letter. You could use N's or something like that. You can use whatever letter you want as long as it's easily distinguishable between the upper and lowercase letter. So some things that aren't good are like O's or S's. That sometimes it's really hard. You have to really pay attention to see what's the capital and what's the lowercase letter. It can be hard to distinguish. So pick letters that maybe the upper and lowercase letter just look different. So I tend to use R's a lot. And so purple is dominant to white. And so when we look at this, we could say then that this purple flower is big R, big R. This this white flower is little r. So now their kids are big R, little r. All right, and so so then when we crossed these two purples, so this purple is going to be genetically identical because it's the sibling and it's the same two parents. So this parent's going to give it a purple gene. It'll have a big R, and this parent will give it a chromosome number 1 with a white gene. And so when you cross these guys, you can get 4 different combinations. So we can do it kind of like foiling in math. So the parent can give, this purple parent has 2 choices, and this purple parent has 2 choices of what to give to their kids. So this purple plant could give this chromosome number 1 on the left with the big R. So let's do this. Big R. And this one over here could also give the chromosome on the left a big R. And it has 2 purple genes, so that would be like this purple flower right here. All right, it's going to appear purple because 2 purple genes will make it appear purple. Or this parent, we can give the big R, and this parent give the little r. So you could have the big R and the little r, and now they have a purple and a white gene. What color is it going to appear? Well, purple is dominant when you have two of them in the same dominant recessive, only the dominant shows up in the same individual. So this is your purple flower. So these guys are going to appear purple. And then we could have, so that's the only combination with this parent giving the first chromosome, number 1. This first chromosome, number 1, can match up with this one or this one. So then the parent could pass on the white. So this one passes on the white with the little r. And this parent, though, could pass on that first purple. And so now it has a purple and a white. There's nothing different genetically between these two with the purple flower trait. So this is also going to appear purple. A purple and a white gene together will make it appear purple. And that's how you get your 3 purples. And then lastly, this white can match up with this white. And the 2 white genes, that the only way you can have the flower appear white is having the 2 white genes. And so so that's exactly what happens here. And so that's how we get our 3-to-1. And those are the only 4 combinations of kids that they could have. So now notice here then when we look at this that out of the 4 chances, this is the 4 combinations that these parents could have, that what this means is every time these 2 parents have a kid, they have a 1 out of 4 chance of having a big R kid and having them be purple. Look at here, there's 2 chances, though, that they could have a purple and a white gene and be purple in this manner. So they have a 50% chance, or 2 out of 4 chance of having a purple kid like this, and then a 1 out of 4 chance of having a white kid. So then if we clump all the purple kids, they have a 3 out of 4 chance, or 75% chance every time that they have a child. They have a 75% chance, or 3 out of 4 chance of having a purple kid versus a white kid. And when he did this experiment, when Mendel did this experiment, he found the same thing all the time. So, but to make this true about the P generation, the F1 and the F2 generation, there had to be a couple of things that were true about it. The P generation had to be true breeding. What does that mean? That means that they had to, when we have a purple flower, that means that both of the genes have to be purple. So true breeding means the genes are the same. So when you have a white flower, both of the genes have to be white. So for purple flowers, you couldn't start out with a big R or a little r. And so that's a characteristic that's true about the P generation. If you make that true about the P generation, then the F1 generation will always have a purple and a white gene, and you'll always get a 3-to-1 ratio in the F2 generation. And it's pretty amazing his work that he did. He got a lot of results when he crossed the plants, and he always got this 3-to-1 ratio every single time as he did that. So that holds true. But the thing is though that he crossed a lot of plants. It's not like he crossed just a few plants, but he always got that 3-to-1 ratio when he did. Alrighty, and I guess this shows you that here. Let me put this up a little bit here. Here's his actual data. So when he crossed purple and a white, and eventually in his F2 generation, he gets 705 purple flowers to 224 white. So look at that. He did like almost 1,000 kids. So he did this a lot. But when you reduce that, that's a 3.15 to 1 ratio. 3-to-1 ratio there. He did it with flower position. Got a 3-to-1 ratio. Seed color, 3-to-1 ratio. So he got this 3-to-1 ratio all the time. And so that's part of his famous work that he did. So going back to our notes here, we can say, and looking at this, some other vocabulary. It says when 2 of the same versions of the gene are present. This is the word homozygous. That probably sounds familiar to you. 2 of the same versions of the gene. So what does that mean? Going back to this, is that when the individual, their genes for that trait are the same. So either 2 purples or 2 whites. And so homozygous would be, let's write this here, big R, or big R, little r. So then when the next one, when two opposite versions of the gene are present, that is heterozygous. And so opposite versions, if we go here, would be having a purple and a white. And so that would be big R, little r, would be an example of that. And then the next word, pairing. So these kind of go together, these kind of go together, and these kind of go together. The genes that an individual has for a particular trait is called their genotype. So that would be, what genes do they have? Do they have two purple genes, a purple and a white, two whites? And so your genotype is this, that you can either be, big R, little r, or little r, little r. This tells you what genes they have. Then from that, you can tell what their physical appearance would be. So if you know their genotype, then for the flower color trait, you could say, okay, if their genotype is big R, that means they have two purple genes and they appear purple. If their genotype is big R, little r, they have a purple and a white gene. Well, I know purple is dominant, so these guys also appear purple. And these have two white genes and they appear white. So the phenotype is the physical appearance. So phenotype, physical, they kind of go together on the F sound, the PH sound. And so phenotype is your physical appearance. So this would be in my example with the flower color trait, is whether or not they appear purple or white. So each individual has, for every trait, a genotype and a phenotype. The genotype tells you specifically what combinations of genes that you have, and the phenotype tells you what your trait is as a result of your genes. And then lastly is we have the word allele. And allele means different versions of the trait. So your allele here, for flower color trait, you have two alleles, the big R and the little r. So for flower color, this is big R or little r. You have two different choices of genes that when you put them together to give you your genotype, you can have different combinations. And so these are alleles. All right, and so those are your genetic vocabulary. So how that relates now to, let's look back at this here, and let's draw the genes in there. And so let's do flower color. So let's do mitosis versus meiosis, relating it to the genetic aspect. So if you had, let's say we're looking at a heterozygous flower. So let's look at a purple flower that is big R, little r. It has a purple and a white gene, but they appear purple. So let's say this is chromosome number 1. So let's say right here is their gene for flower color. And so on that spot, this one's going to have a big R and this will have a little r. So look at what happens now. So when the DNA is replicated. So if we look at this, when the DNA is replicated, this one right here is still going to have in that spot a big R. But remember, when it replicates, this part of the DNA is an exact copy of this part. So this one in that exact same spot, I'm just going to write it above here because it's going to get crowded here. So this had a big R, so if we make it a copy of it, this also has to have a big R. A lot of times people want to put a little r there, and that wouldn't be an exact copy. The little r piece of DNA is right here. This has a little r on it, and so when we make an exact copy of it, this also has a little r. So now when they line up, they condense and line up. So this is going to be your big R chromosome, so I'll just draw a little line right here. This is the DNA that has the big R. This side is an exact copy of it, so that also has a big R. And this one has a little r and a little r. So now go ahead and draw the rest of it in there. So you can pause this and go ahead and draw it. And then I'll go over it. All right, and so as I go over it here, hopefully you paused and tried this out yourself. This chromatid is going to go to the left. This is going to go to the right. So one big R is going to go to the left. One big R is going to go to the right. Same thing with the little r and the little r. So now when they divide and unravel, you're going to have this one having a big R. So this is this one. This one unravels, and it's going to have a little r in that same spot. Same thing goes with these two here. So notice here, now each of them have 2 chromosome number 1s, and they have a purple and a white gene, just like the original cell. So you have clones of each other. And so you have all the types of genes that you have and so you need, and the cell can behave properly. And remember that it would have, in humans, 23 pairs. I'm only drawing 1 pair. So then if we look at how that relates to meiosis, let's do the same thing. So we start out with a cell that is heterozygous, big R and little r. So then, so what I'd like you to do is go ahead, you can pause this and maybe with a pencil go through and see if you can, hopefully you drew this out with me, see if you can trace the genes through. All right, so pause it and do that, and I'm going to go ahead and continue, and you can check yourself. So when this replicates, this big R replicates, so here's the original big R, here's the copy, so right here this is also going to have a big R, and then here's the little r, and this is the original right here strand, so it will have in that same spot a little r, and when it replicates here, this is the replication, we'll also have a little r. So when they line up on the left here, you have the big R chromosome, and on the right you have the little r. Does it matter if, could it line up the other way? Could the little r one be over here and the big R one over here? Absolutely, it doesn't matter. I just chose it to do it that way. All right, and so, so then when they split, the big R chromosome goes to the cell on the left. The little r chromosome goes to the cell on the right. But it's still not half the amount of DNA here because we still have the copies attached at the centromere. So we have to go through meiosis 2, which separates the chromatids. So here, the big R chromatid will go to the right, and this big R chromatid will go, to the left, and this big R chromatid will go to the right. Little r chromatid will go to the left. This little r chromatid will go to the right. And so when that splits here, you're going to have 1 chromosome number 1 with a big R in this cell, 1 number 1 with a big R in this cell. In this cell, you're going to have a little r, and in this cell, you're going to have a little r. And so if you notice here that half the gametes have a big R, half have a little r, which is why when we looked over here, and I said, okay, when you have a big R, little r individual, that they're only going to pass on 1 to their kids, and so they can either pass the purple or the white, the big R or the little r, and the same thing with the other one. And so this is why, because at the end of meiosis you get 4 cells, but half of them are going to have a big R, half of them are going to have a little r, and exactly half the amount of DNA that you had up at the beginning. And not only just half, it has 1 of every kind of chromosome. So either has this one or this one. It's going to give the big R or the little r. And so that's how it relates to genetics and how that works. And so, so if we look over here, so now let's go on with chapter 13. And, whoops, I'm going to write that in with you. Chapter 13 and add on to here. So 13:1, it says, offspring acquire genes from parents by inheriting chromosomes. So we just talked about that, all right? And so in a literal sense, children do not inherit particular physical traits. Literally, they don't. It's not the traits that they inherit. They end up having traits, but they don't inherit the traits. What they do inherit, it is the genes that are actually inherited from parent to offspring. So what are genes? Genes are units of heredity. They're segments of DNA, all of which we just reviewed. Each gene has a specific locus. I want you to know that this locus word means location on a certain chromosome. So that's what I was just talking about. So that's what we did when I drew the piece of DNA and said, you know, there's a specific spot for gene A, gene B, and so on. So they have a specific location on a specific chromosome. One set of chromosomes is inherited from each parent. What does that mean? So one set means that you can only get 1 of every kind of chromosome. So you get 1 number 1, 1 number 2, 1 chromosome number 3, all the way to 1 chromosome 23. So you have 1 of each type of chromosome. That's called a set of chromosomes. So that an egg cell has a set, a sperm cell has a set. When they fertilize, you get a zygote, and that baby has 2 sets, and 2 of every kind of chromosome, one from mom, one from dad. And so we should know that the reproductive cells are called, what is the word for egg and sperm cells? Is gametes. Unite, passing genes on to the next generation. So let's compare. We do sexual reproduction, meaning that you have 2 different parents that contribute genes to their kids. So the genes that the kids get are unique combinations that are different than either one of the parents. So in asexual reproduction, one parent produces genetically identical offspring by mitosis, or if it's bacteria, it would be binary fission. In sexual reproduction, 2 parents give rise to offspring that have unique combinations of genes inherited from the 2 parents. And so that is humans, the unique combinations for this. So this would be humans are an example. In binary fission, we should know our prokaryotes, bacteria. All right, so I'm going to give you some vocabulary terms here. All right, so fertilization and meiosis alternate in sexual life cycles. So a life cycle is the generation-to-generation sequence of stages in the reproductive history of an organism. So let's review and talk about chromosomes in human cells. So each human blank cell, which is any cell other than a gamete, has blank chromosomes arranged in pairs. You should be able to fill that in. Pause it and fill that in. You should have each human somatic cell has how many chromosomes? 46 chromosomes arranged in pairs. We have 23 pairs. A blank is an ordered display of the pairs of chromosomes from a cell. You should know this. Write it down. It is a karyotype. So this right here is the karyotype that we've talked about. So the 2 chromosomes in each pair are called homologous chromosomes, or homologs. So if you see this word, it's the same as homologous chromosomes. Both chromosomes in a pair carry genes controlling the same inherited characteristics, meaning that in the same spot they both have a flower color gene, even though one might have a purple flower color gene and one might have a white. They have different alleles, but the same gene, the same trait, genes controlling the same trait. All right, so here's your karyotype. So you should be able to fill in the next one. Actually, you should be able to fill in the next 3 bullets. So go ahead and pause this and fill in the next 3 bullets. The sex chromosomes are called, you should have X and Y. Human females have a homologous pair of X chromosomes. We have XX for that 23rd pair. So right here is the sex chromosomes in the, right here. This human males have an X and a Y. So for that 23rd pair, they're not homologous. It's X and a Y, so this would be a male. This picture here of the karyotype has X and a Y, so that's a male. All right, so all the chromosomes that have nothing to do with determining sex are called autosomes. So this is pairs in humans, pairs 1 through 22. 1 through 22 have a lot of genes on them to give us all kinds of different traits, but they have nothing to do with determining gender. The one pair of chromosomes that do determine gender is that 23rd pair. So each pair of homologous chromosomes include 1 chromosome from each, what are you going to put there? Parent. The 46 chromosomes in a human somatic cell are 2 sets of 23, 1 from the mother and 1 from the father. And so 23. The number of chromosomes in a single set, so one number 2, 3, and so on, is represented by the letter N. So we have 2 of every kind of chromosome. So we have 2 number 1s, 2 number 2s. So for humans, in most of our cells, it's 2N. A cell with 2 sets is called diploid, or 2N. So the 2 means it has 2 of every type of chromosome, 2 number 1s, 2 number 2s. So for humans, the diploid number is 46. 2N. So we write that 2N equals 46. So what does the 2N mean? It tells us that the 2 means that we have 2 of every kind of chromosome, and that gives us a total of 46. So from this, we use mathematically N for humans, N is 23, because when we have the 1 number 1, 1 number 2, 1 number 3, 4, 5, all the way up to 1 number 23. 1 of every, that's 1 set, but we have 2 of those sets, so to give us 2 of every kind of chromosome. So in a cell in which DNA synthesis has occurred, each chromosome is replicated. Each replicated chromosome consists of 2 identical sister, what do we put? Chromatids. So some of this is review. The only new thing that we haven't talked about before is the diploid and haploid bit. All right, and so when we look at this picture here, this picture shows you a cell that has 3 pairs of chromosomes, and so we have a long pair of chromosomes that look like this. So these guys are a pair, these guys are a pair, and these guys are a pair. And so in this here, it says the maternal set of chromosomes. I'm just going to go ahead and color code them. I use orange. So you get this one from mom and this one from dad. And that's 1 pair. And we know that by the way that they look. And so then you have one, this is a pair. They look identical. And so this is one that you got from mom. This is one that we got from dad. And then here, one that we got from mom, one that we got from dad. And so, oops, I forgot to change the color. One that we got from dad. Okay, and so the paternal, I'm just going to make this color here, and maternal. Paternal means from the father, maternal means from the mother. And so we have 3 pairs of chromosomes. So what do we know about this? There are 3 pairs of chromosomes. And so they're in pairs. So whenever they're in pairs, that tells us that this is a diploid cell. It has 2 of every kind of chromosome. How many types of chromosomes do they have? Well, there's a long one, a medium one, and a shorter one. So there's 3 types. So therefore, N, it will be 3, and we have 2 of every kind of chromosome here that gives us 6. There's 6 total chromosomes, and they're in pairs, so 2N equals 6, and N equals 3, because we have 1 like this, 1 like this, and 1 like this. And so you have 3 types of chromosomes. So that's where this comes from, the maternal set of chromosomes, all the maternal ones, N equals 3, 1 set coming from mom, 1 set coming from dad. So now this baby has double 2N equals 6. And so, they're showing you the replicated chromosomes making it out of the 2 sister chromatids. Okay, so gametes are haploid cells containing only, how many sets of chromosomes does a haploid have? Should have 1. For humans, the haploid number is, what is our haploid number? N equals, you should have 23. Each set of 23 consists of 22 autosomes and a single sex chromosome. So what does that mean? So for an egg cell, An egg cell is going to have one chromosome number 2, one chromosome number 2, one chromosome number 3, one chromosome number 22. But then the 23rd, an egg cell is going to have, it has, for the 23rd pair, it has XX. So it's always going to give an X chromosome. For a sperm cell has one chromosome number 1, one chromosome number 2, 3, 4, all the way up to one chromosome 22. But for the X and the Y is the 23rd pair. So therefore, males, when they make their egg and sperm cells, they can make one with an X, they can make half of them with an X cell, or X chromosome, half of them with a Y chromosome. So this 23rd pair for sperm is either going to be an X or a Y. And so therefore, it just depends. At the end of meiosis, so for males, at the end of meiosis, when you have an X and a Y for the 23rd pair of chromosomes, half the sperm will have an X chromosome, half will have a Y, because they're a non-homologous pair. They're the only pair in humans that are not homologous. And so half the sperm will have an X, half the sperm will have a Y. So in an unfertilized egg, the sex chromosome is always going to be an X. But in a sperm cell, the sex chromosome in the sperm cell can be an X or a Y, and you make them in 50-50. All right, and this is where I'm going to leave off, and we'll pick up on Monday. Have a great weekend, you guys. And I apologize for first hour that this wasn't up and at 'em before you had class. Have a great weekend.